Okay, so next question 141 here. Which of the following statement about lac opera is correct? So this question is asked from lac opera. Here I have keywords is lac opera. Yeah, lac operon. So this lac operon concept from molecular basis of inheritance. Okay, so here there are four options are there. Gene I, GERD, Y, A share single common promoter. Galactose can act as inducer of lac operon. Gene I is constantly expressed. Lactose activate repressor to bind to the operator. So before we make a answer, let's discuss some important things about lac operon. So this lac operon concept was given by two scientists. One is Jacob and Monod. So these two scientists given this lac operon concept. So here the lac operon concept we have like this here. So in this lac operon here we have promoters and here I gene. So here, I gene also called regulatory genes, and again we have promoter and then operator and then JERD YA. Here JERD YA. So these three are structural genes. Here we have operator genes. Here operator and here I gene is a regulatory genes. Here from P means promoters. Okay. So generally what happened here in presence of inducers. So how this lac operon will work we will discuss in presence of inducers. In presence of inducer how this lac operon will work we will discuss. Right. Now generally the main role of this I gene or a regulator gene which undergoes expression means which undergoes transcription process, then it form repressor protein. Repressor here I am writing repressor protein. Repressor mRNA will form, and this mRNA will undergo translation process. Then it form repressor proteins. So like this, repressor protein will form from this I gene or regulatory genes. Okay. If there is a inducers here, if there is a inducers, see this thing it is a inducers. So generally in lac operon inducer we use lactose. So lactose or also we can use lactose and allolactose used as inducers here. So here what happened? So when these inducers are there in presence of inducer only, this operon will work, and here in presence of this inducer and this inducer lactose which binds to the repressor protein. Here this inducer binds to the repressor protein. So inducer here I am writing inducer binds to the repressor protein. So like this bind. So when this repressor protein binded with inducers like lactose, then this whole repressor protein become inactive. Then the repressor protein become inactive repressor protein. So when it become inactive, when this lactose or inducers bind to this repressor protein, then this repressor protein become inactive form. So when it become inactive, so simply we know that. So here this inactive protein which can't bind to the operator. So note here. So inactive repressor protein can't bind to the operator. So when it can't bind to the operator here, when it can't bind to the operator, then this promoter gene which bind with RNA polymerase enzyme. So here RNA polymerase enzyme which bind to the promoter. So this is the concept. So here promoter to the promoter, this RNA polymerase enzyme must and should be bind. So when RNA polymerase enzyme which bind to the promoter, when there is no inactive repressor protein which bind to the operator. Why inactive repressor protein become inactive? Because of inducers. So because of inducers the repressor protein become inactive. When it become inactive, then it can't bind to the operator. When it can't bind to the operator, then RNA polymerase enzyme which bind to the promoter. When RNA polymerase enzyme which bind to the promoter, then these structural genes which undergoes expression, like first it undergoes transcription, then it form mRNA of the respective genes. mRNA form mRNA of structural genes. mRNA of structural gene will form. So when Z gene which undergoes transcription, then it form mRNA, Z mRNA. Y gene undergoes transcription, then it form Y mRNA. Likewise, each and every structural gene which undergoes expression to form their respective mRNA. So once mRNA will form from the Z gene, then this mRNA, Z mRNA which undergoes translation process, then it produce a enzymes or proteins. So that enzymes are called beta galactose. Here I am writing beta. So this beta galactosidase enzymes are produced from the by expression of Z gene, very important. And when Y gene which undergoes expression, like translation, then it produces enzyme called permease. Which enzyme? Permease. When this A gene which undergoes expression, then it produces an enzyme called trans. So likewise, each and every structural gene which undergoes expression to produce their respective enzymes are proteins. So enzymes are mostly proteins, hence. So likewise, Z gene produce beta galactosidase enzyme, Y gene produce permease enzyme, A enzyme is produced trans acetylase enzymes. So all this happen only because of in presence of individuals. Okay. So once we analyze this statement, so here we need to choose correct statement. So gene I and Z Y A share single common promoter. No, this statement is wrong. Why? Because this I gene has its own independent promoter, and structural gene also has its own independent promoter. So this statement is wrong. So galactose can act as inducer of lack of no. Here not galactose. Here who act as a inducer? Lactose. So lactose or allolactose only act as a inducer. So that's why this statement is also wrong. And gene I is constitutively expressed. Yes. So I gene or regulator gene is constitutively expressed to produce repressor proteins. So this statement is right. And here lactose, here see this lactose activate repressor. Here lactose asking what? Lactose which activate the repressor protein to bind to the operator. No, this statement is wrong. Why? Generally this lactose act as an inducer, and these lactose which bind to the repressor protein. Generally this lactose bind to the whom? Repressor protein, not operator. So this statement is also wrong. Okay. So here in which the correct statement is gene I is constitutively expressed. So the right statement is here this one. Okay, clear now? Right. Gene I is constitutively expressed to produce their repressor protein, but here lactose activate repressor, not lactose here inducer inactivate the repressor protein, not activate. So this activate statement make this statement become wrong. So generally lactose inactivate, not activate. Okay, right. So 141 questions, right answer is third one. Next one, 142. Which of the following is used as effective sedative and painkiller for treating post-surgery patient? So here there are some chemicals are given: morphine, antiretroviral drugs, interferons, and antibiotics. Yeah. So these questions which came from the human health and disease. So in this, which mainly frame these questions from drugs abuse. In the drugs, this frame from the opioid drugs are also called morphine drugs. See what exactly the role of this morphine or opioid drugs are, from where we extract these drugs we'll discuss. So generally morphine is a very effective, it is a very effective sedative and painkiller drugs. Pain, and these drugs useful in patient who undergoes surgery. So these drugs use after surgery only. After surgery. Okay, after post surgery. So these drugs used as a sedative and painkiller. Do you know these opioid drugs are morphine drugs which obtained from latex of a white milky like structure, latex of a plant called poppy plant. Poppy plants. So here scientific name of the poppy plant are Papaver somniferum. So in Papaver somniferum, the latex, from this latex of this poppy plant we obtain this opioid drugs are morphine drugs. So here right answer is morphine. So next 143 questions. Which of the following is correct? Order of arrangement of vertebrae column from the head to toe. So here these questions which frame from the locomotion and movement, and this focus on skeletal system. So in skeletal system they mainly focus on like yeah. So axial skeletal system. Axial skeletal system. Yes. So first we need to read the options: cervical vertebrae, lumbar vertebrae, thoracic vertebrae, sacrum. Like that we have four options now. And by this reading these questions, generally this frame from the vertebrae bone. Vertebrae bones. So do you know there are 26 vertebrae bones are present, and all these bones are arranged serially or sequentially only, and these bones are called vertebral column bones. Each bone are vertebral columns bones. So there are 26 vertebral column bones arranged in serially. They're said to be as a vertebrae, or also we can use backbone. So there are 26 backbones are there, and these vertebrae columns bones just now we said they are serially arrange. How they arrange means, so first at the cervical region, cervix region means neck region, we have 7 vertebrae. So and at thoracic region we have 12 vertebrae, and at lumbar region we have 5 vertebrae, and at sacral region we have 1 vertebrae, and coaxial region we have 1 vertebrae. So like that. So it's cervical region and thoracic region, chest region, and then stomach, lumbar region, and then sacral region, and then coaxial region. So like that, the arrangement of vertebrae column bones are serially arranged. All these 26 bones together form vertebrae or backbone. So here we have to choose sequential order from head to toe. So if asked toe to head means we need to go from coaxial bones. From here asking from head to toe. So here first to cervical bone, where we have cervical, we have to choose it. Here cervical, so here sacrum. So we have to remove this by elimination method. Okay, we have to choose this first cervical vertebrae. Here cervical vertebrae. So after cervical we have what? Thoracic. So see here thoracic, no, wrong statement. So here thoracics are there, here thoracic. So we need to remove this. We need to eliminate this two. So after thoracic we have lumbar. So where we have lumbar, see. So here second and lumbar vertebrae and sacrum vertebrae. Okay, na? So this is the right answer. Okay, done. Right. Yeah. Thoracic vertebrae. Yeah. So after thoracic we have lumbar, then only sacrum, then coaxial. So the right answer is in 143. Right answer is second one. Okay. Right. Now we move on. Another important MCQs here asking 140. So here we need to read these questions. Muscle contraction is initiated by a signal, yes, sent by the central nervous system, CNS, by the release of what? So here options are given: cyclic guanine monophosphate, cyclic adenine monophosphate, acetylcholine, acetyl coenzyme A. So these questions also which frame from the locomotion and movement, and here they mainly focus on muscular contraction. Muscular contraction questions here. So in muscle contraction, how muscle contraction will occur, we will discuss now here. So generally here, see, this is what muscle. Just think it is was muscle. So the muscle contain a plasma membrane or cell membrane called here sarco. So sarcolemma is a plasma membrane of muscles. So here we have neurons like this. For example, here I will show you. So here dendroids and cyton, and we have axons. So end of the axons we have axon terminal. Here we have axon bulb. Okay, so this is what neuron. Which neuron here? Motor neuron. So motor neuron transfer signal from central nervous system to the organs. Motor neurons. Motor neuron. See, this is what muscles. So here there is a junction between motor neuron and muscles here. The junction present between motor neuron and muscle here, and this region is called neuromuscular junction. Okay, so here the neuromuscular junction present between motor neuron and muscles. So when muscle want to contract, then our central nervous system which send a signal to the motor neuron, and motor neuron carry that signal to the muscles via here through the neuromuscular junction. Here this axon terminal bulb will secrete a hormone, or they secrete a neurotransmitter hormone. Here very important, they secrete a the information which is carried in the form of neurotransmitters. Okay, so the signal which is sent by the central nervous system via this motor neuron, and this signal is carried out by the neurotransmitters, neurotransmitter hormones, neurotransmitters. So here which neurotransmitter which carry the signal means, here very important neurotransmitter. So that is acetyl. So this generally this acetylcholine is a signal which is carried through this motor neuron, and this acetylcholine which release in these junctions, where neuromuscular junction this neurotransmitter hormone will release, and this acetylcholine neurotransmitter hormone which play important role to contraction of these muscles. So here the right answer is acetylcholine, third one. Okay, fine, right. Next, so next 145. So given below are two statements. One is labeled assertion and reason. Assertion reason type of questions. Here in assertion, four limbs of human and bats are homologous. Reasons: four limbs of humans and bats have similar anatomical structures. Have similar anatomical structures. So in the above statement, most appropriate answer we have to choose. Okay, right. So before that, so these questions which came from the evolution. So in evolution, which is mainly these questions came from the evidence of evolutions. So we have different evidence or proof for evolution. So in that they focus on homologous and analogous evidence. Homo versus analogous evidence. So in questions we are giving about only homologous organ. So we have to discuss both. So here about homologous. So generally homologous organs are homologous evidence in which they perform different function. Very important. So they perform different functions but same origin. They origin same origin, and it is quite opposite to the analogous evidence. So in which they perform same functions. Here same function but origin is different origin. We will take one example to understand what exactly homologous and analogous evidence. So homologous different function and analogous same function, but their origin here same but here different, quite opposite to each other. Here for example, if you take here for homologous, here we have Opensia. For example, take Opensia and mango tree. So generally in Opensia, there is a modification, leaf-like structure. So stem modification is there. That is called phyllo. So phylloclade is a stem modified leaf-like structure. Stem modified leaf-like structures. Phylloclades. And these phylloclades structures which perform a function called photosynthesis, which play important role in photosynthesis process, because in phylloclade means that in Opensia the leaf here in which leaf modified into spine. It means that we don't have any leaves in Opensia, hence the photosynthesis process will perform or carried out by these phylloclades part in Opensia, that is phylloclade is a stem modified part. Okay, photosynthesis. Fine. And see here, and coming to mango tree. So generally in mango tree normal stems. So here normal stems as usual which provide support to the whole plant. So here if you observe this stem and here also stem, both phylloclades also stem part in Opensia. Here normal stem in mango. Origin is same origin, but they perform different function. Here in Opensia, phylloclade perform photosynthesis function. In mango tree, the normal stem which provides support. See, this different function, but their origin is same. Such type of organs are called homologous organ, and these homologous organ leads to divergent evolutions. Very important. They leads to divergent evolutions. Okay, right. Now here coming to same function and different origin called analogous organ. So if here we take one example, like here also we take two organ plants like Opensia. We need to take Opensia, and also we take another plant Bogan. So if you look at these two important structure, so in which same function but different origin. Okay, no, fine, right. So in bougainvillea we have hooks or spines. Hook or spine. So generally this hook and spine, it is a modified form of stem. It is a stem modification which origin from stem. Fine. And if you observe the Opensia here, in Opensia we have spines. Spine is a modified form of leaf modification structures. Leaf modification. Here spine and hooks are spine in bougainvillea. They perform same functions. Here these two spines and hooks are spines in Opensia and spines in bougainvillea. They perform same function. What is that function? Protection from herbivores. Yes, and see their origin is different. Why? How means Opensia is origin from in Opensia spine origin from leaf modification. The origin from leaf. In bougainvillea, the spine's origin from stem. Means here different origin, but they perform same function. So this type of modification is called this type of organs are called analogous organs. Okay, and these analogous organs which gives evolution like convergent evolution, which leads to form convergent evolution. Very important, convergent evolution. So likewise we have evolution homologous and analogous. If you read this statement, and one more important example for homologous organ, here I am explaining you. So for example here four limbs. So example for in mammals like mammals include like whales, blue whales, and bats, and cheetahs, and humans. In mammals, whales, bats, cheetahs, and humans, they share similarity in the anatomical structure. So here, anatomically they same, means origin is same. The origin same, but they perform different function of their four limbs. How means like whales use four limbs for swimming, and bats use these four limbs for flying, and cheetah which use 4 limbs for running, and a human being use these 4 limbs for walking, like locomotion. So like that, see, the origin is same, structure anatomically the origin is same, but they perform different functions of this 4 limb. So this is also one of the examples of homologous organs. Okay, so by this analysis we can, once we analyze this assertion reason type, 4 limbs of humans and bats are homologous. Yes, this statement is right. And here reason is 4 limbs of humans and bats have structural similarities. Both have structural yes, both are having structural similarities, means their origin same. So origin is same, only they differ in their functions. So both assertion and reason both are true statement. So both A and R correct. R is the correct explanation for A. So right answer here, third one. Okay, so this is all about the homologous and analogous evidence organs. Okay, and remember, homologous organ which leads to divergent evolution, and analogous organ which leads to convergent evolutions. So here Y means they function differently, here they functions like same function they perform. Convergent means same, divergent means different functions. Okay, done. Right. Okay, next one, 146. Given below are two statements. Statement type of question. In statement 1, Down syndrome. Here Down syndrome is caused by the absence of one of the X chromosome, and statement 2, Turner syndrome is caused by the presence of additional copy of chromosomes. So here these questions which frame from the principle of inheritance. These questions frame from the chapter principles of inheritance. Principle of inheritance. In this chapter they mainly focus on chromosomal disorder. Okay, so here first we will analyze, we have to give some explanation what exactly this. Here keywords are Down syndrome and versus Turner syndrome. We will discuss Turner syndrome. First, we will discuss about Down syndrome. Down syndrome. So here, generally, Down syndrome is a disorder of autosomal disorder. It is a auto disorder. Remember, it is a autosomal disorder. So this Down syndrome is due to, it is occur due to presence of one additional copy of 21st chromosomes. Additional copy of 21st chromosomes is leads to cause Down syndrome. Okay, so Down syndrome. So here how means like this. So generally it is an autosomal disorder. Do you know, in human chromosomes there are 23 pair of chromosomes we have. Yes, human being has 23 pair of chromosomes. So among 23 pair of chromosome, 22 pair of chromosomes are said to be as a auto, and only one pair of chromosome are said to be as a allosomal. Allosomal are also called sex chromosomes, and these allosome as sex chromosomes are only differ in male and in female. In male, this allosome chromosome is XY. In female, the allosome chromosome is XX. Remember. So here, this Down syndrome is due to Down syndrome which occur only in autosomal chromosome, means 22 pair of chromosome, 1 to 22. So in 22 pair, so where exactly this Down syndrome will occur means at 21st chromosome. Generally 21st autosome, how they are arranged means like AA means 21st chromosome, like this. So from starting from 1 to 22 we have all autosomal chromosome only. So at 21st autosomal chromosome, actually every chromosome has in pair. Yes, but here what happened, one extra autosomal chromosome is added. One extra autosomal chromosome is added. So then here we have 3 copies. So instead of 2 copy of chromosome, here we have 3 copies of autosomal chromosomes, and these 3 copies of chromosome are said to be as trisomy. Okay, so these 3 set of chromosomes is nothing but Down syndromes. Okay, so generally at 21st chromosome there must and should be present only one pair of chromosomes, means 2, but here one extra chromosome is added to the 21st, then it leads to cause Down syndrome. Fine. Right. Now coming to Turner syndrome. Turner syndromes. Here Turner syndrome is a, generally it is a allosomal disorder. Very important. Allosomal disorder means sex chromosome disorder. Allosomal or sex chromosome disorder. So this disorder which occurred due to absence of one X chromosome. Here absence of one X chromosome. And how means, for generally this Turner syndrome which occur in female only. Very remember, which occur only female. How means, generally female has 23 pair of chromosome, in which 22 pair of chromosomes are autosome, one pair of chromosome is allosome, and female has allosome chromosome XX. 2 XX chromosomes generally they have. So here what happened, Turner syndrome, 44 autosome chromosome is there as usual, but here only we have only X chromosome, but another X chromosome is absence or loss of one X chromosome or absence of one X chromosome. Then here we have total only 45 chromosomes. Generally 46 chromosome must and should be present, but here due to absence of one X chromosome, here one X chromosome, it become 45 only, and this leads to a disease in female. That syndrome is called Turner syndrome, and it is a this disorder which occur only in sex chromosomes or allosomal disorder. Okay, right. So this is all about the Down syndromes and Turner syndromes. Okay, and we need to recall some important autosomal disorder, more autosomal disorder. Not only Down syndromes, there also we have Edward disorder, Edward syndrome. So generally this Edward syndrome occur, it is also autosomal disorder which occur at the 18th chromosomes. So generally 18th chromosome has one pair, but here one extra chromosome is added, like that, 18th of try condition. And here important is Edward syndrome occur due to addition of one extra autosomal chromosome to the 18th place. And one more we have, that is Edward and Patu syndrome. Sorry, Patu syndromes. So this Patu syndrome is also autosomal disorder like Down syndrome. And here also addition of one extra autosomal chromosome at the 13th chromosome. Generally 13th chromosome has only one pair, but here one extra autosomal chromosome added, then it become trisomy. Here also we have three chromosomes at 13th place, and here 18th place, and in Down syndromes 21st place. So we have to remember, so in this all only autosomal chromosomes only get extra chromosomes, only autosomal chromosomes only get extra or additional chromosome to the pair chromosomes. So then it leads to cause some autosomal disorder like Down syndrome, Edward syndromes and Patu syndromes. But here Turner syndrome occur only in sex chromosome. And we need to remember one more allosomal disorder which is occur in male. So in male, allosomal disorder, allosomal or sex chromosome disorder, that is Klinefelter syndrome. So generally in this Klinefelter syndrome which occur in male, so in male, here I am writing, so this Klinefelter syndrome which occur due to addition of one X chromosomes, due to addition of one extra X chromosome. Remember, here deletion of one X chromosome from the female, but here addition of one extra X chromosome. Generally male has 44 autosomes and XY, so that is 46 chromosomes are present generally. But in Klinefelter syndrome what happened, one extra X chromosome is added. How means, like 44 autosome plus X here Y, double X Y. And if you count this, total number of chromosome is 47. So in Klinefelter syndrome we have 47 chromosomes, in Turner syndrome we have 45 chromosomes. So here one deletion of X chromosome, here one addition of X chromosome. So deletion of X chromosomes, that leads to cause Turner syndrome in female, and addition of one X chromosomes, it leads to cause Klinefelter syndrome in male. So this Turner and Klinefelter both are allosomal or sex chromosomal disorder, and here Turner syndrome is due to monosomy condition, and this Klinefelter is due to trisomy condition. Trisomy 2n plus 1, here 2n minus 1, monosomy. Okay, done. So this is all about the important concept. Here if you read this statement, so we will do, so I will drop this one, just wait. So once we read, so which statement is correct statement, yeah, here we will go. So here Down syndrome is the caused by the absence of one of the X one. Generally Down syndrome is not related to the sex chromosome, so this statement is wrong. So statement, wait here, yeah, so here this statement is completely wrong. Why? Because Down syndrome is a autosomal disorder, not allosomal disorder. So this statement is wrong. And here Turner syndrome is caused by the presence of additional copy of chromosomes. No. So it is the due to, and it is this Turner syndrome is allosomal disorder. Generally it is allosomal and here autosomal. So this statement is also wrong. And Turner syndrome, how it caused, we will discuss here. And here Down syndrome, it is not the due to absence of X chromosome, and it is not due to the Turner syndrome is not due to the additional of copy of X chromosomes. Okay, so these questions completely mismatch, means like completely opposites. So here in this contest both statements are wrong. Okay, so here we choose. So here statement 1 is correct, no, and statement 1 is correct, no. This one, third option, both statement 1 and statement 2 are correct, no, and both statement 1 and statement 2 are incorrect. So right answer is fourth one. Okay, so next one, 147. So which of the following statement about the reabsorption process in Henley's loop are correct? Here there are four options are there. So one is descending limb of Henle loops is permeable to water but almost impermeable to electrolyte. Urine gets concentrated in Henle's loop. Reabsorption of sodium and water take place in Henle's loop. Active or passive transport of electrolytes occur in the ascending limb of Henle. So choose the correct answer. Here we have to choose correct answer. Okay. So let's start some explanation regarding this Henle's loop. So generally these questions which is from the human excretory product or excretory system. So in this completely focus on nephron, and nephron means urine formation. So in nephron they focus on one important part, that is Henle's loop. So here we need to draw only Henle's loop. So generally nephron has various important parts. So here we won't discuss each and every part. Just we focus on Henle's loop here. So here I am drawing. So generally, so Bowman capsules. From this Bowman capsule, PCT, proximal convoluted tubule. Then we have descending limb of loop of Henle. U-shaped structure is called loop, Henle's loop. And then sorry here, and then yeah. So like that we have nephron structure. So in the nephron structure we first identify the okay. So in this nephron structure first we identify the part. So we know that here Bowman capsule. So Bowman capsule, in this Bowman capsule there are different arterioles together make some small capillary like structure called glomerulus, and Bowman capsule plus glomerulus together form a structure called here Bowman capsule and glomerulus together form glomerular apparatus. Okay, and here see part of nephron, renal tubule part. In that first one is PCT, and this part from here U-shaped structure called Henle's loop, and here DCT part and this CT or conducting tube part. Like that we have various part in nephron. So here in these questions they frame only this loop, Henle's loop. So in Henle's loops here this region is called, here I'm writing with another pen. This part is called descending limb of Henle. Here descending limb, and this part is called ascending limb. So Henle's loops has two limbs. One is descending limb and another one is ascending limb. So what exactly happened here in a descending limb of loop? This descending limb of loop which is permeable to water. Here water is permeable. So here permeable to water, very important, permeable to water. So descending limb permeable to water, but it is im to electrolytes, very important, electrolytes. So remember, descending limb of loop of Henle permeable means allow water, and it does not allow impermeable to electrolytes. Here electrolytes and water, and it is quite opposite in ascending limb. See here. So in ascending limb what happened? It is permeable to electrolytes, permeable to, here I am writing electrolytes, but it is im to water, remember. So as fluid flows down in the loop of Henle, so water moves out. Generally what happen? Permeable now water, so water moves out. So when water comes out from this loop of Henle, then here in this loop of Henle nephron tube there is a concentrated urine will be formed. Concentrated urine will get formed. Okay, so all these are about the and importance of this loop of Henle. We will go through here. Here see this, the descending limb of Henle loops is permeable to water. See, descending limb of Henle loop permeable to water, yes, but almost impermeable to electrolyte. Right, this statement is right. Okay, no? Correct statement. And here urine gets concentrated in Henle's loop. Okay, so we'll discuss later. First read this. See, reabsorption of sodium and water take place in Henle's loop. Yes, almost. Why? Because this ascending limb permeable to electrolytes. Okay, and this statement is also right. So active or passive transport of electrolytes occur in the ascending limb of Henle loops. Yes, this statement is also right. So here urine get concentrated. Generally urine get concentrated in this collecting tube. In collecting tube only the urine get concentrated. But here urine get concentrated in limb of limb. So this statement make this wrong. So answer is A, C, D. So here we have fourth options. A, C, D is the right one. So next one, 148. If the diploid chromosome number of typical angiosperm is 36. Here, these questions which is from sexual re in flowering plant. Okay, in which they frame from diploid mathematical. Okay, questions. This question is applied mathematical type of questions we will do. Okay, yeah. So in questions what they are asking, if the diploid chromosome number if the diploid chromosome number of typical angiosperm is 36, here 2n, how much? 36 diploid. What would be the chromosome number if it is in its endosperm? So let's start how endosperm will form. Generally in angiosperm, in angiosperm the endosperm form by triple fusion. Very important, triple fusion. How means? Generally in angiosperm, in embryo sacs we have, see, this is embryo sac, here we have eggs and here we have central cell. Okay, fine. So here we have two gametes, two male gametes, in which first male gamete fuse with egg cells, So here first male gamete fuse with egg cell. Fuse means fertilization, then it result to formation of zygote. So zygote has 2n, diploid number of chromosomes. Fine. And what about second male gamete? So this second male gamete which fuse with central cell. Here second male gamete fuse with central cell. Here central cell has two nucleus, and male gamete always has single nucleus. So the fusion or fertilization between second male gamete and central cell, then there is a formation of 3n triploid number of chromosomes. The structure is called endosperm. So like that endosperm will be formed, and this endosperm has 3n number of chromosomes. Okay, so in this angiosperm two times fertilization will occur. The first time fertilization is called syngamy, and second fertilization is called triple fusion. So syngamy and triple fusion, two times the fertilization which occur in angiosperm, hence the angiosperm fertilization is called double fertilization. Okay, done. Right, first fertilization syngamy, second fertilization is triple fusion. So triple fusion, second fertilization which occur between second male gamete and central cell has two nuclei. Here two nuclei, so there is a formation of 3n triploid number of chromosomes. Our structure is called endosperm. So once we analyze, so what is here asking, so diploid chromosome number of typical angiosperm is 36. What would be the chromosome number in it is endosperm? Simple. So if it is become haploid, for example, here we will do 2n is equal to 36. If we want n, then 36 by 2, then 18. So here n 18 is the what? N number of chromosomes. But here asking 3n. So why? Because endosperm has 3n triploid chromosome, na? So 3 into n what? How much n? 18. 3 into 18. Okay. So here 18 into 3. So 8 3 is 24. Yeah. Yes. 24. 2 3 1 5 50. So right answer is 54. So 3n contain 54 chromosome. Okay, fine. If they asking only haploid means, then it become 18. If triploid, then it is become 54. Here doesn't mention triploid chromosome of endosperm. We know endosperm contain 3n number of chromosomes. How means like this. Okay, so right answer. So 148 right answer is first one. So that it means 54 chromosome it contain. Okay, so next one 149. Arrange the following in descending order. Here descending order of number of species in the Amazon rainforest. We have to identify the number of species in descending order. Okay, fine. And here we need to know descending order means from highest to lowest. We have to identify highest to lowest only. Okay, so here there are various species are given in Amazon rainforest. So like plants, birds, fishes, invertebrates and mammals. So choose the correct answer, and that must and should be from descending order, from means highest to lowest. So once we recall, so in Amazon forest rain in Amazon, so these questions which is came from bio and its conservation. So in which they mainly focus on one important forest, that is Amazon rainforest. In the Amazon rainforest has the highest biodiversity. Has the highest biodiversity in the world. Highest biodiversity are present in this Amazon rainforest. Okay, fine. So in that there are highest number of species are invertebrates, in which highest species are invertebrates. How much means 1,25,000 of species of invertebrates are present in this Amazon rainforest. So then plant species. How much means 40,000 plant species are present in this forest area. And then after fishes. So how much means 3 species of fishes are there. And next birds. So how much means 1,300 species of birds are present in this Amazon rainforest. And next one is mammals. So here mammals, just 427 species of mammals are present in this Amazon rain. Okay, no? So likewise, so there are various huge biodiversity are seen in Amazon rainforest. So here we have to arrange this in descending order, means highest to lowest. So highest is invertebrates, lowest is mammals. So here choose this answer first, invertebrates first. So here invertebrates means D first. Then after invertebrate we have plants. Second, after plant we have fishes. Third, so fishes after birds. And at last here we have mammals. So arrange this one. So D first option is must be D. Here just wait. Yeah. So first one is D, and here second where we have, yeah, plant A, and the third here where third fishes C, and fourth, what fourth, B, birds, and the last we have mammals. So the right answer is fourth one. Okay. Right. Next 115th questions. A population of diploid organism is at Hardy-Weinberg equilibrium. So here Hardy-Weinberg equilibrium. So these questions which frame from the principle of inheritance. Principles of inheritance. What? Hardy equilibrium. So here, if the frequency of allele A is 0.1, here the frequency of dominant allele, here asking A is dominant, capital A. So here asking about frequency of dominant allele, that is capital A, 0.1, and the frequency of double A. So we have to find out here the frequency of capital A. So how much? So here we need to apply an equilibrium, Hardy-Weinberg equilibrium equation. So that is P square plus 2PQ plus Q square. Okay. So this is the equation for and equal to 1. So in which here dominant allele expressed by P and recessive allele is expressed by Q. So here asking about P. So here the P is 0.1. So we have to find out capital A, means P square. So 0.1 into 0.1. So here answer is 0.001. Sorry, not double zero, only single 0.01. So here we have to find out only this P square. Okay. So why? Because so P here dominant allele 0.1. So here asking to find out the frequency of capital A, means individual, in which P square is equal to 0.1 into 0.1, 0.01. So right answer here 0.01. Okay. So likewise we have to find out the dominant, the frequency of individual genotype, that is dominant, represented by P square. We have to find out P square here. And one more important MCQs here. Yeah. So the opening between the right atrium and the right ventricle is guarded by. So these questions which frame from the body fluids and circulation. Body fluid and circulation. So in that mainly focus on internal structure of heart. In internal structure of heart, they focus on valves. Here asking about valves. So here, if you draw the diagram of heart like this, here, so sinoatrial ventricular here. So these two are upper chamber. So upper chamber of the heart always receive what? Blood, which receive blood. And these two are lower chamber of heart, and this lower chamber of heart always pump the blood, pump or send the blood out of the heart. These lower chambers are called ventricles, and these upper chambers are called atria. So this side we have right side, and this one left side. Remember, upper chamber there is atrium, and that is right side always carry deoxygenated blood from various tissues, and this left side of the chambers always pump oxygenated blood. Here oxygenated blood means the blood with oxygen, and deoxygenated means blood with carbon dioxide. So like that we have, and here and this one right atrium, and this one left atrium, right ventricle and left ventricles. So here the atrium and ventricle both are separated each other with the help of a septa called atriosepta. So this atrioseptum which divide the heart into upper chamber and lower chamber. So here only we have seen valves. So there only we have walls. There are two walls are present. So the right side walls and left side wall. So the right side wall which is present between right atrium and right ventricles. Here left side the walls present between left atrium and left ventricles. So here the walls which is present between right atrium and right ventricle is called tricuspid wall. Very important. Tricuspid wall. Here we have tricuspid wall like this. So here we have tricuspid wall. And the left atrium and left ventricle, here also we have seen one wall are called bicuspid wall. So these bicuspid walls are also called mitral wall. So here we have left atrium, and between left atrium and left ventricle we have bi wall or mitral wall. So once we analyze the opening between right atrium and right ventricle, here asking about right atrium and right ventricle, which is guarded by which wall means tricuspid wall. So here answer is tricuspid wall. And here bicuspid wall present here between left atrium and left ventricles. Okay, which is present between left atrium and left ventricle. And a sinoatrial node present where? Here in right side of the sign, where it is present? Right side of the right side means right corner of the right atrium. And semilunar walls, the walls which is present in the blood vessels. So which is present generally in the blood vessels. So here we have systemic semilunar wall and pulmonary semilunar wall. Like that we have various. So like that the 151st question's answer is fourth one. The right answer is fourth one. Now coming to which of the following enzymes synthesize precursor mRNA. Here precursor mRNA. So 150 we will discuss. 152. Yes. So here these questions which is framed from molecular basis of inheritance. So in which especially focus on RNA polymerase enzyme in eukaryotic organism. Generally in RNA polymerase there are three different types of enzymes. There are three types of RNA polymerase enzyme. They are RNA polymerase 1, and second one is polymerase 2, and the third one is RNA polymerase. So like that three RNA polymerase enzymes are present. Now we will discuss what they transcribe or what they synthesize. So here, sorry, okay, done. So here this RNA polymerase 1 enzyme which transcribe or which transcribe or synthesize what? Ribosomal RNA or RNA. In ribosomal RNA we have different types of ribosomal RNA. All type of RNA synthesized except to 5S rRNA. Very important. 5 here 5S rRNA except to 5S rRNA. All type of RNA are synthesized by RNA polymerase 1. So like they synthesize 28S rRNA and 23S rRNA and 18S rRNA. Like that each and every ribosomal RNA synthesized by RNA polymerase 1 except this 5S rRNA. So then this 5S rRNA synthesis by RNA polymerase 3. Here I am writing. So 5S ribosomal RNA synthesized by RNA polymerase 3. Remember. Okay. And not only here 5.8 S rRNA also synthesized by polymerase 1. So these are the four important rRNA which synthesis by RNA polymerase 1, but except 5S rRNA. Here synthesis. And coming to RNA polymerase 2. So which synthesize first hnRNA. So this hnRNA was the pre for messenger RNA, mRNA. Messenger RNA. Fully matured RNA is mRNA. So after fertilization, after transcription, this RNA which undergoes some modification. So before modification, the RNA is called hrRNA. hnRNA means heterogeneous nuclear RNA. hnRNA means heteronuclear RNA. So after transcription, then it form hnRNA. So this hnRNA undergoes post-transcription modification like capping, splicing, and removing introns and exon. All these things post modification of transcription. Then it become formed into mRNA, messenger RNA. Since messenger RNA is a precursor of hnRNA, and these two RNAs are synthesized by the RNA polymerase 2. Now coming to RNA polymerase 3. So here RNA polymerase 3 which transcribe or which synthesize very important tRNA, transfer RNA or adaptive RNA. And not only tRNA but also synthesis 5S rRNA, and also it synthesis snRNA. So these three snRNAs, tRNA and 5S RNA synthesis are transcribed by polymerase 3, and hnRNA and mRNA are transcribed or synthesis by RNA polymerase 2, and very important, ribosomal RNA synthesis by RNA polymerases. All type of ribosomal RNA synthesis by polymerase 1 except this 5S RNA. So all these are about RNA polymerase enzyme in eukaryotes. So once we read these questions, which of the following enzymes synthesize precursor mRNA? Here precursor mRNA, what? Precursor mRNA, that is nothing but hnRNA. So which enzyme synthesis this precursor RNA, hnRNA, RNA polymerase 2. Here right answer is RNA polymerase 2. So fourth one is the right answer. Okay, right. Okay, next one, 153. Given below are two statements, statement type of questions. One is labeled as assertion, reason type of questions. So read assertion. In recombinant DNA technology, that is RDNA technology, means these questions which is framed from bio and biotechnology. This question framed from principle and process. Biotechnology, principles and process. Okay, right. So here about RDNA technology, recombinant DNA technology. In recombinant DNA technology, lysozyme, very important enzyme here. Keyword is lysozyme, is used for disrupting bacterial cell wall. Yes, so this lysozyme used to destroy or to break the cell wall of bacteria, while cellulase is for plant cell. Yes, here cellulase enzyme which used to destruction of cell wall of plant cells. So this statement is okay, fine. Isolation of genetic material needed disruption of cell. Yes, so assertion and reason both are a true statement. So once we discuss why this RDNA, in RDNA technology we need this lysozyme cellulase enzyme. And do you know, in biotechnology process, in RDNA technology process, there are various steps are involved. In that, first and most important step is isolation of genetic material. So here our genetic material is DNA. So we need to isolate DNA from the selected cells, either it is bacteria or plant cell or any other cell. For example, if you see where this DNA is generally present, any cells, the DNA is present in the nucleus. How means in nucleus we have chromosomes. Now these chromosomes are generally made up of with DNA and histone proteins. So what we need to do, we need to break all these membranes. Yes, all these membrane to isolate this DNA from the nucleus. So first of all we need to break cell wall. We need to break or we need to disrupt the cell wall. So here we, if using plant cell, if we need to isolate DNA from plant cell, then we use a cellulase enzyme for destruction of cell wall. Okay, and if we want to isolate DNA from bacteria cell, then we need to use an enzyme called lysozymes enzymes. The lysozymes enzymes, this one. And here cellulase enzyme used for cell breakdown of or degradation of cell wall of plant cell. If we want to isolate DNA from fungi cells, from fungi, then we need to use chitinase enzyme. So like that, based on various cells, we use different types of cell wall degrading enzymes. So for plant cell we use cellulase enzyme, for bacterium cell we use cell lysosome enzyme, for fungi we use chitinase enzyme. So all these enzymes which play important role for the destruction or for the breakdown of cell wall. So once cell wall get breakdown, then again next step what we need to do, we need to break the cell membrane or any new cell organelle membrane. So for breakdown, for disruption of this cell membrane or cell organelle membrane, then we use detergent lysis. So like that we have to disrupt each and every membranes. So finally we get our desired isolated DNA. So like that we need to isolate by breaking all this and a component which is present in the cell, especially their cell wall. So here this question which is framed from how cell wall get breakdown, why cell wall get need to break, means to isolate our genetic material. Definitely there is a need of destruction of this cell wall of cells. Okay, so likewise both assertion and reason type of questions are correct. Here both A and R correct. R is the correct explanation for here. So right answer is third one. So next 154, which of the following hormone is not secreted by human placenta? So these questions which is framed from RE. So here generally placenta develop in female body during pregnancy. So when it develop during pregnancy in female body, and these placenta act as a endocrine part. It act as a endocrine part. So it is act temporarily. So only during pregnancy. So once pregnancy is over, then this placenta also disappear. Okay. So likewise, placenta act as endocrine tissues during pregnancy only. Remember, this placenta act as endocrine part only during pregnancy. And during pregnancy, this placenta play important role to secrete various hormones. Especially there are four important hormones which is secreted from this placenta during pregnancy, and they are especially one is H hormone, second one is H hormone, and the third one you know that estrogen hormone, and also fourth one progesterone, estrogen and progesterone. So these are the four important hormones which is secreted from the placenta because of they act as a temporary endocrine part during pregnancy. So here HCG means human chorionic gonadotropin, human chorionic gonadotropin hormone, human chorionic gonadotropin hormone. HPL means here human placental hormone, and estrogen and progesterone both are the this estrogen progesterone also secreted by the ovary. This also secreted by the ovary during menstrual phase or during the development of follicles. During development of follicles, this estrogen progesterone also secreted from the ovary. So like that, there are four important hormones secreted from this placenta. So here we need to choose. So not, here asking about not. So highlight this word not, secreted by the human placenta. See, so here progesterone secreted by the human placenta, human chorionic gonadotropin hormone secreted by the placenta, estrogen also secreted by the placenta. So here not is LH. So why means this LH luteinizing hormone secreted by, which is secreted by anterior pituitary gland, secreted by the or from the anterior pituitary gland only. Not only LH, along with FSS also secreted from the anterior pituitary gland. They also play important role during oogenesis, means development of ovary. Oogenesis, development of ovary or ovum. Okay, so likewise, so not means here right answer is second. So next 155, colostrum, very important. Colostrum secreted by mother during initial days of lactation and is abundant in, so which antibodies which abundant in colostrum milk. So these questions also frame from RE. So let's discuss some things about colostrum milk. Colostrum, generally colostrum is a yellowish fluid which is secreted by mother during initial days of lactation or immediately after birth of baby. After birth, in initial days, this fluid will be secreted. That fluid become yellowish. The yellowish fluid is called colostrum, and these colostrum are rich in antibodies. It's rich in one antibodies, or also we can use immunoglobulin. So here this immunoglobulin antibodies are nothing but IgA, very important. So IgA has abundant during in colostrum milk or yellowish fluid which is secreted by mother during initial days of lactation only. So IgA is the right answer. IgA. Okay, right. So this IgA immunoglobulin or antibodies provide, this IgA provide passive immunity. Very important, which provide passive immunity to child to protect the new infant from various pathogens, and it provide immunity or passive immunity to the child to protect the newborn infant. So very important, IgA, and it provide passive immunity. Remember, provide passive immunity. Okay, right. So next 156. So read this statement. Consider a population of 10 million cells. In a population there are 10 million cells. Given the per capita birth rate, so that is 0.02. So birth rate, how much? 0.02. Birth rate is 0.02 per unit time. And the per capita death rate also given, D. Death rate, how much? 0.02. The expected number of cells after 10 generation. So expected number of cell after 10 generation. So like that we have questions, and these questions which frame from the organism and population. So in that topic, which is mainly focused on growth rate, we discuss some explanation on this birth, death, and after 10 generation what happened. And here you need to know that population growth, so is the intrinsic rate of natural increase. So in which natural increase? So here intrinsic rate of, here I am writing in rate of natural increase, that is R. As so we can measure population growth as an intrinsic rate of natural increase as R is equal to birth rate minus death rate, or simply we can write R is equal to B minus D. Okay, so here where here R is intrinsic rate of natural increase, so here R is equal to 0. Okay, so here what we need to learn here from here, and we need to do R is equal to how means birth minus death. So here 0.02 birth rate and 0.02 death rate, so equal to R. So R is equal to 0. So here this where R is equal to 0, which represent what means the population growth rate becomes 0 now. So it means that population size remain completely constant. So it means that, so when the population growth rate here, when the population growth rate become 0, that is R is equal to 0, it means that the population size remain completely constant. Okay, so regardless how many generation which pass the population, like 10 millions or 20 millions, so regardless this generation, if population growth rate becomes 0, means it the population size remain constant only. So the right answer is only here 10 millions. Why? Because same as it is, population size must and should be constant. So hence the right answer is 10 millions. So next one, given below are two statement 1, plasmids are autonomously replicating DNA. Read statement 2, plasmids are extra chromosomal DNA. So in the here we need to choose most appropriate answer from the option given below. So these questions which is framed from bio and biotechnology, they focus on tools of RDNA technology. In tools of RDNA technology, we use one tool, so that is vectors. So mostly we use plasmid as a vector. So about plasmids, generally we know that plasmids are circular and double-stranded DNA, circular double-stranded DNA, and it is an extra chromosomal DNA, extra chromosomal, and it has self-replicating nature. Okay, so these are the characteristic of plasmids. Generally plasmids are present in prokaryotic organism only, that is bacteria. So in bacteria, for example, if it is a bacteria, so in bacteria itself has the main genome of bacteria here, bacterial chromosome or main genome are also called genome. Along with this bacterial chromosome, there are extra chromosome, and it is circular and double-stranded DNA is present. And this DNA has self-replicating nature, self-replicating in nature. This DNA is called plasmid, and this plasmid used as a vector in RDNA technology. Okay, right. So here plasmids are autonomously replicating DNA. Yes, this statement is true statement, means correct statement. And plasmid are extra chromosomal DNA. Yes, this statement is also true, means correct statement. Both statement 1 and statement 2 both are correct. So here statement 1 is correct, wrong one here wrong, and third one, both statement 1 and statement 2 are correct. So right answer is third one.